Okay, James, the screens are yours.
Well, thanks everyone for showing up. It's nice to see you guys in some sense, at least see your names on my screen. Today I'm going to be talking about one of my research or one of the chapters in my thesis using the series Jaguar methodology, which many of you have already been exposed to, I'm sure. But here I'm applying it to the G2F hybrid data set. And we'll go over what series Jaguar is, and then I will go over my results. So as you all know, the phenotypes that we observe in the field are a product of genetics, the environment, and their interaction G by E. And plant breeders have traditionally used joint regression analysis, which is often called Finley-Wilkinson regression, to visualize and understand G by E. And this is a simple linear equation where the phenotype of the ith genotype in the J environment is equal to some intercept, which is just the mean performance of that genotype, plus the slope for that genotype, which is the response to environment, multiplied by the observed environmental means plus a residual. And in the bottom left, I have a toy example. You can see the red line here has a slope of 0, so it's not responsive to the environment. The green and blue lines have a steep slope, so they are responding to environmental factors. However, the blue line has a low mean and the green has a high mean. In 1966, Eberhard and Russell looked at joint regression analysis, and they pointed out in it that it would be desirable to replace this IJ term, this environment mean, with an environmental factor. And that, in short, is what the series Jaguar methodology is doing. The series aspect of it identifies the environmental factor, and then the Jaguar is a genomic prediction part of it. So series stands for critical environmental regressor through informed search. And as I just mentioned, its objective is to create an environment index to use in place of environment mean. And it does that by iteratively looking at the correlation between different environmental parameters and the environment mean. Jaguar, joint genomic regression analysis, uses genomic information to permit the prediction, that permits a prediction of untested genotypes, and it does it by estimating a slope and intercept parameter. Again, going back to the linear equation. I applied this to the genomes to field hybrid data set, the 2014-2015. We can see is a relatively unbalanced design from panel A. Most hybrids were evaluated in about 5 to 8 environments. However, we do have a handful that were evaluated in about 28, 27. We're going to look closer at panel B in the next slide. So we'll just skip over that and look at C and D, which are showing how diverse the environments are. Panel C is the cumulative GDD for the first 100 days after planting. So we can see in the coolest environment, there's roughly 1,500 GDD accumulation, and in the warmest environment, there's about 2,500. So that's a wide range of temperatures that these hybrids experienced. And panel D is at day length. And we can see that in these southern locations, they experience a much shorter day length, not quite approaching like a tropical day length, but there's still some diversity in day length. And then the geographic distribution you can see on the map on the right. I believe the northernmost location is South Dakota. And then we go as far south as Georgia and Texas. So when we zoom in on the PCA, well, we can see that it is divided into 5 major testers, LH82, LH185, LH198, PHB47, and PAC51. So we ran the analysis for the entire hybrid data set, which is 715 hybrids in 31 environments. And then we also subsetted that for these 5 testers. We looked at 6 traits, 2 flowering time traits, stage 2 anthesis, stage to silking, 2 height traits, plant height and ear height, and 2 grain traits being grain moisture and grain yield. The genomes to field locations have a weather station within the field. which we use to create our environmental parameters, or excuse me, our environmental factors. Any missing observations we filled in using DayMet. And we looked at the following 5 environmental factors: day length, GDD, photothermal time, which is GDD multiplied by day length, photothermal ratio, which is GDD divided by day length, and diurnal temperature range, which is the maximum temperature minus the minimum. And some of you may have seen this animation or these slides I'm going to show before, but I think they pretty clearly explain the series process. So for each environmental factor, we look at all windows of time starting at days after planting. So first, let's say we look at 1 to 7 days after planting. We calculate a correlation between the environmental index that was accumulated between 1 to 7 days and the population mean. And then we visualize that using a little heat map here on the right. We go to the next window of time, which here we're just saying is 1 to 13 days after planting, calculate a correlation, fill in the heat map, and we do this over and over again until we find the maximum correlation with environment means, which here is 0.996. So then we would use that environmental index in place of environment means. So now we're going to look at the window, the environmental indexes that series found. Solid lines here are days to anthesis, dotted lines days to silking, with GDD being the environmental factor. And we can see that all the windows of time overlap, which is a positive. We would expect GDD to be the environmental parameter, since corn growth and development is driven primarily by temperature. What's also encouraging is that it appears that these windows of time overlap with the V6 stage of development, which is a particularly important time in corn growth. It's when corn is transitioning from vegetative to floral growth. But we cannot definitively say that, since that was not phenotyped in the population, but it is just encouraging that it occurred in this time. For plant height and ear height, plant height being solid line, ear height being the dotted line, photothermal ratio was the environmental factor for most of the subpopulations, as well as the entire genome field data set. However, there's some variability. For example, the LH185 uses diurnal temperature range for ear height, and LH198 uses GDD for ear height. But all in all, we do see the windows of time overlap. So we can say it appears that the same environmental factor is driving or can be used to predict plant height and ear height in this data. But as the trait complexity increases, we get more variability in the environmental factor and window of time. Grain moisture, the solid line, is relatively conserved in terms of days after planting, and with about 2 populations using PTR and the rest using GDD. However, for grain yield, the most complex trait, the window of time is spread out, and there's no real conserved environmental parameter used. I think some of you also have probably seen this slide, sort of reviewing the prediction scenarios that we employed. We could say that 1 to 2 is predicting tested genotypes in untested environments. I prefer to call that TGUE instead of 1 to 2, but I believe every slide has it written both ways. 1 to 3 is predicting untested genotypes in tested environments, UGTE, and 1 to 4 is predicting untested genotypes in tested environments, UGUE. We use a tenfold cross-validation where 90% of the genotypes are used to fit the model, and they are used to predict the other 10%. Just looking briefly at the results for the entire G2F data set, our columns are the prediction scenarios 1 to 2 in the first column, 1 to 3 in the second, and 1 to 4 in the third. And then we have day strength thesis in the first row, plant height, and then grain yield. And for the simplest trait, day strength thesis, in all prediction scenarios, we achieve pretty good results. That's sort of expected, you know. We should be able to predict antithesis by now, and we do. We do a very good job even in the most complex scenario. We see for plant height and for tested genotypes and untested environments, we still obtain a relatively good overall prediction accuracy of 0.48. Even in the 1 to 3, we get 0.56, but by the time we're predicting untested genotypes and untested environments, our prediction accuracy does start to slide. For grain yield, we do obtain satisfactory prediction results for 1 to 2, but as the prediction scenario becomes more complex, our prediction accuracy starts to decrease, as well as the predictions start to cluster together. There's not much separation between the environments, which is unfortunate, but it's, to some degree is expected as well. Grain yield is difficult to predict. This is an overview of all the traits and all the subpopulations. You know, the black bars are the 1 to 2 to tested genotypes and untested environments. The medium gray bars in the middle is 1 to 3 untested genotypes and test environments, and the light gray bars on the right is 1 to 4. So for all the data subsets and the traits, they're all relatively the same, with one notable exception, which is the LH198 subpopulation. Has higher prediction accuracy for all 3 prediction scenarios for the more genetically complex traits, most importantly being grain yield. The average prediction accuracy of the other 4 tester subsets is 0.47. But then for LH198, that increases. I have written down 0.59, but that is clearly above 0.59. So we are encouraged by the results for LH198. LH198 was grown in a narrower range of environments, which we think helped account for the high prediction accuracies. It was grown more in the Corn Belt and less in Texas and Georgia. Siri Jagger was able to achieve modest to high prediction accuracies for all traits and data subsets. And as you can, we're looking at the LH198 untested genotypes and untested environments. The overall prediction accuracy is 0.71. We have a good separation of environments. Looking at the within environment prediction accuracy, you know, we have some very good environments like Ontario 2014 has a 51%, Wisconsin 14, 53. But then to be fair, we also have some environments where there's something else going on. Missouri in 2014 has a negative 0.1. New York 14 has a 0.04. So we have some environments where we get very good prediction accuracies for grain yield, and other environments where there's something to be desired. With that, I would like to thank everyone in my lab group, everyone in the G2F initiative for collecting all this data and making it publicly available. Sources of funding, USDA, the Iowa Corn Promotion Board especially, they were the first funders of the G2F initiative, the P3 Plant Phenomics Group, and the NSF. So with that, I'll take any questions.